going to talk about mitosis and the cell cycle. And really what we're getting here is cell reproduction. So we have two objectives. One is that you'll be able to identify phases of the cell cycle. And these mitotic stages, you'll be getting a description, just here's what they do in words. But also we do graphic representations where you'll actually see pictures of them. Sometimes they're pictures such as drawn by an artist. So some definitions to start off with here. First, somatic cells. Somatic cells are your body cells. These are every cell in a human body except for in females the egg cell and in males the sperm cell. And these are produced through mitosis. Simple division of the current existing cells then result in having these somatic cells. And we have 46 chromosomes in the nucleus of every single cell. And so these 46 chromosomes are actually 23 pairs. So what I mean by this is we have chromosome number 1, chromosome number 2, down through chromosome number 22. And then you have an X chromosome and a Y chromosome if you're a male, or an X chromosome and another X chromosome if you're a female. So when you get down to that, you have 46 total individual chromosomes, but you have the 23 that are pairs. So the genes that are on chromosome 1 are the same on one copy of chromosome 1 as they are the other copy of chromosome 1. You may have a different variation of that gene. For instance, a variation could be you have one with a blue eye allele and you have a brown eye allele, but even though they are not identical, they both are the gene for eye color. Okay, so that's what that is getting at. And these two different copies of the chromosomes are called homologs. Okay, and so homologs are each member of a chromosome pair. So you have two chromosome 1s, and they are homologs. We are diploids, which means we have a total number of 46 chromosomes in all of our cells for the majority of our lives, and that with the exception of our zygotes, which zygotes are sex cells. So then we have haploid, so here's the cell cycle, okay? The lifetime of a cell. And we've already looked at this, and so this will give you kind of an idea of what we're talking about here. So here you have the orange circle, and that has that I there for interphase. And then you have 3 phases within interphase, or 3 sections of interphase. G1 is where the cell is really only doing the duties of the cell. In the S phase, new DNA is synthesized to start getting ready for mitosis. And then G2, more processing to get ready for mitosis occurs. Then you have mitosis. That's the M. It's purple in the inner ring and yellow in the outer ring. And in mitosis, you use about 5% of a cell's life cycle. Okay? And 95% of the life cycle would be in interphase. So, in interphase, growth stage 1, main thing, metabolic activity of the cell. That's when we're saying the cell's doing its thing. The scientific way of saying it is that metabolic activity of the cell, whatever the cell is needing to do. Synthesis stage, Metabolic activity of the cell continues. The cell continues doing its thing, but you also have replication of DNA. And then growth stage 2, where you have metabolic activity of the cell, and the final preparations for division in mitosis occur. Mitosis. It happens in all cells at some point. However, some cells rarely do it. And at some point they could do it, but may never do it in their lifetime within an organism. However, some cells don't do it as often. Cell division process. There's 5 major stages. And here in this picture, you can see cells that belong to all of those stages. So the first one, prophase, is the nuclear envelope disappears. Okay, so that the membrane that surrounds the nucleus disappears, and the chromosomes get real thick and condensed. That's why we can see them once we get to prophase. When we are at in the interphase sections, we are not seeing chromosomes, and that is a key to you. Oh, this is an interphase, and you can see the sister chromatids and the centromere. The spindle also starts to form. Next, metaphase. The chromosomes move to the equator of the spindle, so they start to centering up, okay? And then in anaphase, the centromere split into 2. And you'll see that picture with each sister chromatid being pulled back to the polar ends of the cell. And these sister chromatids are pulled apart to opposite poles of the cell, and each chromatid is now a separate chromosome. And then in telophase, the nuclear envelope starts to reform. So you'll start seeing a newly divided cell with 2 nuclei, and chromosomes start to uncoil again. So now they're going to not be visible again once telophase occurs. And finally, cytokinesis. And this is where the cytoplasm itself divides. So when we talk about prophase, metaphase, anaphase, and telophase, we're talking about the nuclear